Hello. Good afternoon. Welcome back from lunch. I hope everyone is awake. Otherwise, there's coffee. There's coffee outside for sale, which you can patronize. Okay, so this afternoon, I am truly honored to be introducing the second keynote speaker for today. Professor Basiru Bomfo trained as a veterinarian, but he also holds a PhD in epidemiology. He worked for a while with, I think, the Veterinarians Without Borders, and then worked as a postdoc fellow with the Swiss TPH for about 4 years. And then he moved back to Africa, and worked to establish what we know as the One Health concept right now. I can't even begin to tell you how instrumental he's been trying to bridge the gap between the North and the South, and also to foster South-South collaboration within Africa. What I'm most proud of this gentleman is he is the one who gave me my first break into life back after a PhD in the West. When I finished my training in Tennessee and I moved back to Ghana and was hungrily looking for a job, he interviewed me over Skype. I was about 7 months pregnant then, with my face all puffy. And I told him that, well, this is my situation, and he asked me whether pregnancy was a disease. I said, no, it's not a disease. As long as you be able to deliver on the job that you are applying for, by all means. And so...
Merci beaucoup, Lydia. Très belle introduction. Je pensais que tu allais la faire en français. Comment est ton français? Okay, good afternoon. What Lydia just said, I could have just stopped my talk asking you some question around what we did. We just stand here and... So thank you very much. As she said, I'm from Togo, and I could have finished my career without knowing what is happening in Ghana. It's just 20 to 25-minute flight from Lomé to Accra. But some of the initiatives gave me this chance to interact directly and indirectly with Ghana, and even beyond Ghana and Togo, from Cote d'Ivoire to East and West Southern Africa. So before I start my talk, I just want to recognize the power of the Deltas program. And I don't know if Kevin Marsh is in this room. Just want to acknowledge his contribution for the design of this program, and also thank Suzanne, she's there, who's really supporting us running the Deltas program. I don't know if Denise is around. Denise? Yeah, thank you very much. As she said, I'm from Centre Suisse de Recherche Scientifique in Côte d'Ivoire, which is a regional research center. With the name, you may think that it's Swiss, but it's an African institution. We just kept the name Swiss for the branding, because when you hear Swiss, then you don't even ask yourself, you can give money. And I'm the director of the consortium Afrique One, and Afrique One is one of the consortium like WACCBIP. And we run that with other 14 fellows. And the 14 fellows of Deltas, I think Gordon is here, they decided to put in my hand hot potatoes. Just to coordinate, I will not say the mess, but the most difficult part of coordinating the health research of 14 brilliant leaders, research leaders. And you have heard yesterday with what have been said about Gordon and his colleagues' work here. So this is what we call ASH, the African Research Consortium on Health Research. I don't know if I have. So what I'm going to talk this afternoon is just to give you an insight of what Deltas has done for the last 6 to, I will say, 10 years, and bring to your attention one of the concepts or the approach we have developed, which is One Health. And I will define that for you to understand. And since yesterday, I have been listening to most of the talk, and when I see what is happening in the lab with nice pictures from the lab, I just ask myself, what's the meaning for a farmer in a village? And what could be the impact of that for a farmer or a woman who is selling food at the market? I don't say that basic science is not important, it's very important, but sometimes we have to stop and see what are the pathways to the impact on the life of people. So everything started for me when I did my DV, and I said, so on which topic? And then I read the application, and they said, you choose the topic that is relevant for Africa. We don't want to guide you on which topic you want to do. This is where I also call Lydia saying, okay, I take you as a postdoc, just tell me what you want to do. I give you money and you can fly. So this was what we call the African Institution Initiative. I will not come back on the reason why research support or capacity building is very important. It was to handle the transition period for young PhDs and to make sure that we build the environment for young researchers who have finished their research to come back. And by that time, the most difficult part of our institution was that we did not have a postdoctoral position in our universities. This was the first time we tried to introduce, and we faced a big challenge at the universities asking us, where do you want us to put those fellows? They are not recruited as assistants, and they are just hanging like bats, if I can say. They are not attached to that. And some of them were saying, no, we want this position. We said, if you want this position, you need to reduce the time of lecture, because this program is for science. You need to take 80% or 90% of your time for science. So this was the big challenge, and we were happy that just after, I think Gordon knows the system to understand how best Africa can be supported. And this is where in 2016, together with the 11 consortium, we decided with the support of the funders to build the ASH, this think tank program that I'm coordinating. So ASH is made of 20 consortia coming from I think 16 to 17 countries, and we have about 100 researchers that are part of this network. But the 14 directors of the consortium form the ASH, and we are established to support all funders, to support all kind of research challenges in Africa, because if you look at the group across Africa, we claim that we have the legitimacy to talk about what is health research in Africa. Just taking WACCBIP here, if you see the impact of WACCBIP and you multiply that by 14, you will see how the impact is in in Africa. We also help the funders to understand how, where to target investment and where to build the capacity. And the proof of concept of deltas is to shift the center of gravity of science or research into Africa, but not only research, but the funding mechanism and the management of research. This was the challenge that we had also for the trust from the funders to give this amount of money to African institutions without any good governance system. And then we have to look at areas of building the capacity. The capacity was not only for fellows, but it was also for institution and administration, and especially the financial officers. And you will see later, I will tell you, it's not only science capacity that we need, but we need to build the capacity of other part of the management program. And the model is based, as I said before, on the African defined priorities. What are the main priorities? Supporting institutions and people within the institution, and using also the tools. And if you look at this graph in blue, you see the program looking at different capacity building aspects. And we have some programs who help to develop tools, those programs who are deeply into the health research and health priorities. And this was something very important. And when you look at the 11 or the 14 consortia, you may ask yourself, is there competition among ourselves? But for us, it was not competition. It's to look where we can cross-fertilize, have synergies, and share platform. Sometimes when I'm here and I see fellows from WACCBIP, they think that I'm part of WACCBIP because of we were just exchanging. And when Gordon go to Cote d'Ivoire or wherever, and the fellows see them with Lydia, they think that we are he's from Africa One. But for us, being part of one or another is not the important, but it's how we share resources, how we share skills. This is the most important part of the business. So what have we achieved so far? As I said, we moved from 11 to 14 consortia. And when we look at the figures, in 5 to 6 years, it's more than 2,000 fellows trained by the Deltas program in Africa, with more than 1,000 research networks and 5,500 undergraduates. And if you look at the network, you will see how our network is established. And you may see that the challenge is in Central Africa. And that was something Deltas was thinking, and for the current phase, we made possible to embark institutions from Central Africa into the program. It's 1,400 publications that we had. And if we take the funds leverage on top of what we did, each of the consortium mobilized 3 to 4 times the grant. I had 6 million euro for Africa One as Aspire. And we, at the end of the program, we were around 36 million, because this grant helped us to attract more grant by our fellows. And we have very strong institutions. And what is very important for this program is the balance between sectors and the balance between countries. Because sometimes when you build capacity, an ivory tower of an institution or center of excellence somewhere, and other countries or institutions are not following, it's a big problem. And you see what's happened. I'll just give you the example of Sierra Leone, Liberia, and Guinea with Ebola. Without capacity building in that area, when a disease emerges, you have it on your face. And to tell you, for example, today in Liberia, you have, I think, 1 or 2 vets in that country. Only 2 veterinarians, and they are occupying the high-ranked political position. So in the ground, you don't have people. So you see, if you take that into research, you can have very strong institutions doing research, but if you have weaker institutions or weaker countries, it's a problem. So Delta was trying to establish this network to help. And we have contributed during the COVID, I will not come back to that, how science can contribute to better prepare pandemics. And we had a paper on that. And we have also developed through the case of 4 consortia, how to really monitor and evaluate capacity building. We can monitor and evaluate research, but monitoring and evaluating capacity building was a big challenge, and we tried to put some tools and methods for that with the help of our partners in the North. And that gives us now a very good system of improving the evaluation and setting targets. The challenges, you all know. I remember when Lydia wanted to go to a conference, I think that was the first time I realized that I have to pay for the baby, for the flight. I have to recruit a nanny during the conference for the child there, and I have to squeeze the program so that there are very specific time there that she can work. Just to give you an example. And sometimes funders also focus on the micromanagement, what I call micromanagement. Managing funding, good managing funding is good. But sometimes we forget to think that research evaluation is also very important. And the last thing I want to highlight is the mobility of researchers. Visas. It's a nightmare. And I don't know, maybe Gordon, we need to do something. We have been talking about that. This is where we need institutions like ECOWAS or African Union to help. I cannot understand that if I want to go to Kenya today, I have to take 2 weeks of application of visa, not knowing if I want to go or not. They cannot call me on emergency on something in a country where I have the skill and I can decide the following day to go. It's happened during COVID and Ebola, where when we wanted to bring East African social scientists to West Africa, it took a lot of time. So those kind of things are challenges, and we need to think of that. And one thing I want, as a mutual learning, is the area of administration overheads. And sometimes we struggle with funders. And the second element is keeping the funding foot in Africa, because we lack science funding mechanism in Africa, and we still depend on external funding. And what I want to highlight here is the freedom to look at or to address African priorities, and especially health problems that Africans face. And now I am going to tell you the example of the One Health approach that I sold in 2008 when I was applying for that grant. So the One Health approach and how it can help the fellows here and even practitioners. So One Health approach, if you look at Africa today, we have more than 100 epidemics, outbreaks ongoing. I don't know if in Ghana here you have the one dengue. Do you have dengue here? Ongoing dengue. So what are we doing on dengue? And you look at those kind of diseases that we can see, we listen, but there are others that we don't even look at. Food poisoning, antimicrobial resistance, malnutrition, mental health problem, those kind of health that every day affect people are not considered. And if you look at the health threats as a complex system, we have two ways. Either we use our tool, the tools we have in hand, which is not effective. And on top of that, you can also have the different perspectives, how we see the problem. The perspective of the communities, the perspective of natural science, and the perspective of humanities. So the most important things that One Health address is the collaboration between sectors, between disciplines, and then looking at the knowledge generated by this system to transform the knowledge with the contribution of the communities. So this is what we call the transdisciplinary approach. And why One Health? Because we see that if you look at the biology, you look at the physiology, you look at the pathology, human and animal share the same ecosystem. And there's no difference of paradigm between the physiology or biology of human or animal. They share the same pathology. And if we want to handle some of the diseases, we have to look at both sides to understand the issue. Then we ask ourselves 4 main questions. And the first one is how those threats emerge and how, for example, the pandemics are fabric. And what is the cost of those pandemics or outbreak on the the livelihood of people. And the third question is, what is the capacity of our institution, our health system to handle them? And the fourth question, do we have innovative solutions or approaches to address them? And if we look at the emerging diseases, some of the diseases from animal to human, we can definitely see that it's possible to handle them in the livestock sector to prevent human health. And I give you this example so that you see. This man is a physician. We were working in the northern of Mali. And if you want to look at the health of this child on the medical perspective, it's okay. And then another component will be what will be the behavior of this woman to accept the type of medication you bring for the family. You can stop there. But with the One Health concept or approach, you can understand the health of this child by looking at the livestock, the source of their nutrition, which is milk. And some will stop with the nutrition, but those will go even far away looking at the grass available for livestock, which increase the milk production. So if you want to look at the health of this population, this pastoral population, and you only look at the child, you will miss a lot. So it's important to look at the global perspective of the environment of the patient, because everything is interlinked. Then comes to the failure to look at this holistic approach. You fail to look at this holistic approach, and if you look at the incidence cost with the time, this red curve, you will see that when an outbreak emerges, the cost is very high. We have estimation on that. If you look at Ebola, if you look at COVID-19, the cost of COVID-19 was very high, but it was possible in the wildlife sector, in the domestic animal sector, to make a surveillance and detect and respond quickly. If that was done, we could have reduced the cost. And the objective of the One Health is to flatten this red economic costs by using different mechanisms. We have the International Health Regulation that sets 19 priorities, the performance of veterinary services, and then looking at the condition with our contact with wildlife and the wash area to prevent the disease before it becomes a big problem. And I give you an example. Since we are here in WACCBIP, you work on AMR, antimicrobial resistance. And if you take a chicken, I think we had at lunch chicken. And if you take one chicken sold in the restaurant, the farmer gets from the price you have on the chicken in the market, the farmer gets 13% of the share. 13% of the share. But who do we ask to make effort to reduce antimicrobial use? Is the farmer. So if you put a social scientist there, what will be the willingness or the motivation of this farmer to prevent the health of people at the expense of his or her business? This is where the challenge is. The same thing happened with milk. Look at what farmer get from 1 liter of milk per day. This is the case in Mali. And if that farmer use antibiotics in 1 or 2 cows in the farm, and you ask the person to not use or sell this milk, if you have 5 liter or 10 liter per day, you see the amount of money the person will lose. So bringing this perspective to the problem will help to see how best we can improve the condition, the livelihood of people in order to protect the human health. And One Health is really relevant to Africa due to lack of resources, and we have demonstrated through different studies that you gain 14 out of 1 investment in using One Health. And one of the main problem now, if we take rabies, we know that rabies vaccine is effective, very good vaccine. It can protect, we can stop the transmission of rabies by vaccinating up to 70% of dogs. But now the problem is not in science, it's in decision. So we can move from the social science up to the political level on decision, how to make decision on, and I can give you more example on the added value of the One Health. So this is also one illustration of One Health. I brought this from Jürg Usinger from Swiss TPH. You see how our health system function with mass drug administration every year. It's good for business to sell the drugs, but all the time, every year, people get reinfested. So what do? Do you only rely on mass drug administration, or you need to add sanitation, good quality water? So those kind of things we are improving. And with that, we have trained many fellows in One Health, and we have a massive open online course on One Health you can attend. Every year we organize that in October, and you can attend this course with coaching in October, but out of October you can still attend, and but without coaching. And we can get a certificate at the end of the training. My last message is this picture. The first one representing, I will say, maybe fellows who want to capture everything. We are not really focused. We need to focus. And it also represents researchers in Africa. If you are a researcher, it's not like in Europe you have your lab and you know what to do. In Africa, you have to handle administration, you have to handle social life of people, you have to handle many, many other things. This is a researcher. And at the end, they will evaluate what is your impact in science. And the second message is supporting women in science. This capacity building is also there. When the women are showing their steps to grow, we need to look at social barriers that are really bringing them and breaking the glass ceiling. And the last one, I don't know if you realize, the seniors sometimes are not very good in helping younger ones to grow. This, Gordon, am I right? Sorry. I missed the acknowledgement. This one is very important. What happened? The last slide. Okay, I will leave you with the conclusion, is this one. Those are the champions. I think Gordon is our leader in this. I'm coordinating, but we know that when we look how things goes, is the real leader, and I thank you very much for inviting us to here.
Thank you very much. Thank you very much for that wonderful keynote talk. In fact, when he was talking, two things came to my mind. The first one, for those who are biochemists here, is a signal transduction cascade, right? Phosphorylation and dephosphorylation. One small grant opportunity from deltas, or should I say one big, has evolved to something massive. And now we have this arch platform, which is fantastic. And the second thing is really about the crosstalk between disciplines when we are conducting the kind of research that we do. We tend to focus too narrowly on only the area that we are more important, should I say, intellectual in. But I think we need to start talking across disciplines and formulating our research questions more broadly, so that when we are done, we'll be able to communicate the science that we have done to the right people who need to hear it, whether it's policymakers or even the communities or even educators. So with that, I think I would like to open the floor for questions. We have some time for questions. Yes, at the back.
Yeah, sorry. Thank you very much. So I believe you spoke on capacity building, but are you also looking beyond capacity building, transferability in science research? So beyond just doing the research and bringing the research to life, are you also looking at that? You also spoke on a bit on communication, science communication. There are times that I've interacted with people and then they complain that, scientists, the way we explain things to the communities, the lay people, it's a bit technical. So what advice would you give to like young upcoming scientists in that regard?
Thank you very much. Very good question. I went very quickly. This is part of what we call the soft skills. But beyond the soft skill, we have one scientific foundation of One Health is the transdisciplinarity. What we do, and sometimes we have problem with funders on that aspect. When you recruit a fellow in Africa One, the first thing we do, I think we have Africa One fellows here, some of them are here. The first thing we do is tell you, go to the target population, not only the communities, but those who are going to take your research, like the national program. If you are working on TB, you need to go to the TB program and talk to them, talk to the communities. This is the starting point. Don't wait until you finish to start engaging them. And there's in Delta's program, what they call capacity community engagement grant that they put on top of the Delta's initiative. But in our program, Africa One, we already have that, which sometimes is not seen as purely science, but which help for policy and communities to take what you bring. If you wait until the end and you come with your result, the probability that is taken is very, very low. And we know the adoption rate of any innovation, the adoption rate is very poor. So if you want to increase the adoption rate, is at the beginning you have to set the research question with the people. And the second aspect of what we call transdisciplinarity is when you work with other disciplines and you generate knowledge. You generate knowledge from biologists, from chemists, from whoever, and you have a scientific knowledge. The second step is to put that knowledge into the community so that you take also the communities or the decision.
And also that WACCBIP epitomizes. In your experience, is this culture widespread at the moment? That's my first question. And secondly, is that for Africans in diaspora who are still thinking of some way of engaging with back home, are there other avenues aside the Delta program and others? Are there other avenues where that's available at the moment for those who are still contemplating coming back? Thank you.
Yeah, it just remind me one researcher in Switzerland saying, I want to work in the area where there's no malaria. And I said, in Ivory Coast, there's nowhere you can have yeah. So we have problem. The context of Africa is problem, we know. We also have potential. But here what we advise, nobody will, at the beginning, this country. But I came back and I started setting my One Health lab, and you see. Today, I don't know which country to take. I just take the country that I love, like Ghana, but I'm in Ghana, I will not say that. If you ask the Malian government where to put money, and you put research and to buy arms, what are they going to choose? I think it's the priorities, but it's our responsibility to show that science can help to handle some of the problems. And we need to show them also, or to discuss with them on different mechanisms of funding. For example, in Cote d'Ivoire, we have tried, I was explaining to Kevin Marsh yesterday, we try to set up a national science foundation. What do? We had the debt given back from Switzerland to Cote d'Ivoire. And we said, okay, we want one share for science. And they said, no, we cannot prove that this money will really yield development. And we said, okay, we want this money, but it's your own money, and we have we put it as an endowment fund. And the interest that is generated every year is used to enroll 16 to 20 fellows, masters and PhD fellows in the country. And after 10 years, when we have evaluated, the country realized that the number of fellows we are training is gaining in momentum. And when we had the evaluation, the Ivorian government decided to put another $12 million into that. So doubling the portfolio of this grant. And then we have now an established science foundation where we ask funders to put money into that pot, but not diversifying it. And we call it what the wedding gift scenario. Do you know that? If you are having a wedding and you ask people to bring you gift, you may end with maybe 10 coffee machines. But if you have a list in the shop, when somebody pick a coffee machine, so you'll have to choose something else. So we have to bring funders to choose areas where we still have problem. Money is there, but money is going in the same place.
I think we have time for, just one more question. And the mic is already, okay, well, maybe two. Yes.
All right, good afternoon. Okay, so I'll start with our sister's question about science communication. By way of that, we have a show called Science Digest Show, of which Prof. Gordon Awandare champions for us as well. So we want to say thank you to him. We talk about science and health. We try to demystify science and health over there to the populace. So you can subscribe on YouTube, Science Digest Show. I have a question about collaboration, Prof. Yesterday, Prof. Ernest Aite spoke about collaboration. You also talking about collaboration efforts here. I want to know, how challenging has it been? Could you share some practical experiences about how to go about that so that we can leverage on the strength of each other to push the scientific community forward? Thank you.
I think collaboration is a culture, and we have not been taught how to collaborate. Our academic system is built in a way that we don't talk to each other. Look at how faculties are set up, and sometimes we don't have inter-faculty collaboration. And this is what we are building now in the academia. And my first PhD student who did the work on One Health with collaboration between different sectors, when he wanted to defend the thesis, it was almost rejected because they said it was not science. They don't know from which discipline he was. Those are the kind of thing we have as a problem. But it's this kind of research that can solve the society's problem. Not only a single discipline knowledge. A single discipline knowledge cannot solve a problem unless you mix that and then look at the area. And for the communication, I agree with you. We need to learn also how to communicate. But communication starts not on only one way. As I said, it's a transdisciplinary process. It's an iterative process. And I have seen that now there is a big interest of companies, small companies in science communication, which that help to take science to the society. And it's a very big component in our system now. And if you look at how others communicated on COVID-19, they were even stronger than scientists. So we need to make efforts in that.
Whilst we are on the topic of science communication, if you all have your phones, please go to the WACCBIP handle. There's actually a graduate student of WACCBIP who is a science illustrator at the same time. And for every single talk that you've heard, she has illustrated this very, very beautifully. Take a look at it and please retweet. This is ScienceCom live as it goes on. Good. We have one more question and then I'll end the session.
Thank you, Prof, for your beautiful presentation. My question is related to the last picture. The senior who is cutting the scale to go up, how will he get back? Don't answer that. But what can we do?
How?
Repeat.
Should I repeat?
Yes.
I said the last picture.
Yes.
We had a senior going
who's
up
cutting the steps.
and cutting the steps. I was asking how will he get back when he got all the way to the top. But my reflection point, please.
You know. Thank you.
Prof. Bofo, please.
He will fall. He will fall.
How?
That's all. That's a very simple.
How can we change the mentoring culture in Africa. It is very important. You're giving funds to us to do research, but we are confronted to very poor mentoring. We have to humanize the mentoring in Africa, and I think each consortium has to take this into account. Thank you.
Yeah, I think it's a good and valid point. We take that into account, and it's part of monitoring and evaluation of capacity building. So being a good scientist, I think yesterday it has came, being a good scientist is not only the number of papers. We want to know how many fellows you have trained. Not only train, what are they doing? And when you move, who are at your back? Or when you retire, who will take over? And sometimes we have some of seniors retiring without anybody taking over. So those are the kind of thing we should put in the evaluation system of research.
Okay, so I'd like us to please acknowledge and thank Professor Bonko. Thank you so much. And next, we are going to move on to the plenary 7 and the session 3. The title of that session is Drug Discovery Mechanisms and Resistance. I'd like to call the session chairs Dr. Evelyn Yaria-Boni. Is it from? Did I miss it? And then Sebastian Arthur. Thank you. Thank you very much.
So I want to, even as we have session chairs, I just want to steal their job a little bit to introduce the next plenary speaker. I've known Thibaut since 2011. We're in the same lab. And now we are collaborating on a grant that he's leading. So I thought it was very apt that he would come and share some of the work that he's been doing with us. So sorry, Evelyn and Sebastian, but I'd like to call Thibaut to present his talk.
Thank you. That's a tough act to follow. We are going to go from the broader perspective to the very narrow perspective of HIV drug resistance, but I hope you will find it interesting. Thank you. So I'm going to have a fairly technical talk about HIV drug resistance. If you feel that you fall asleep or something, you can stand and walk around. And I will focus on a specific drug which is called dolutegravir. So dolutegravir is an integrase strand transfer inhibitor, and it's now part of the WHO antiretroviral drug regimen recommended for everybody. And one of the reasons for this broad recommendation is that essentially it's very, very difficult for HIV to become resistant against dolutegravir. So we say it has a very high barrier to resistance. And we did actually a fairly a good amount of work with Peter, including on this topic particularly. And so that's one of the stones that we put in this wall. We studied indeed the barrier to resistance of dolutegravir, and it's indeed very hard for HIV to evolve resistance against this drug. And one of the reasons is that the resistance mutations that are coming up against dolutegravir have poor fitness, and so they are poorly replicative, and they have low levels of resistance, which led us to talk about an evolutionary dead end for the virus, whereby HIV cannot evolve high fitness, high resistance viruses when people fell with dolutegravir. However, we also had concern that maybe we're missing out on something, because this is true for the results when we sequence integrase, which is a target gene of this integrase inhibitor. In the Domono clinical trial, so Domono was a clinical trial that was led at Erasmus University, and the idea was to test the current regular antiretroviral regimen and compare it to dolutegravir monotherapy. Because dolutegravir was performing so well that people thought maybe we can just use it as a single agent. It actually turns out that you cannot and you should not, but in the course of this trial, one of the patients who fell was actually developed a mutation in this, what is called the 3' PPT region. And you can see this change. Well, you cannot see it, but you have to trust me. It's published, so. And there is this G stretch in the 3' PPT that was changed. So, of course, I'm sure a lot of you don't know what the 3' PPT is, and that's where it's a bit technical. So 3' PPT plays a role in the reverse transcription of HIV. The reverse transcription of HIV looks complicated, but it's very simple. You have the initiation here with a tRNA primer, and then you have the production of this strand of DNA from the RNA template, and then you have what's called a strand jump, and then you have production of another piece of DNA that is illustrated here. And during reverse transcription, another thing happened, which is a degradation of the RNA template. This is done by RNase H. And the 3' PPT is actually this part here in blue. It is resistant to the degradation by RNase H, and therefore it's a piece of RNA that remains after the synthesis of the other strain. And from this RNA can start to act as a primer for the production of the other DNA strand, which is this one. And then there is another jump, and you get eventually a double-stranded DNA that gets integrated in the human genome, and there you have a real infection. So the way we understand the 3' PPT mutations is that because this region is mutated, it cannot serve, it's not resistant against RNase H anymore, cannot serve as a template for the initiation of the second strand of DNA, and therefore we have the creation of circular forms of DNA called 1-LTR circles, which are therefore immune to integration inhibition because they simply don't integrate, but they readily can lead to the production of new viral particles. So the integration process is bypassed by these mutations, and therefore dolutegravir should not be active anymore. The only question, of course, is that this is true for some of the 3' PPT mutations that have been discovered or explored in vitro mostly, but not for the mutation that was found in the Domono clinical trial. So at this point, we still don't know, are these 3' PPT mutations a real problem, and do we need to monitor these mutations in people who are failing dolutegravir, as this medication is rolled out now on the continent, and that is used for nearly 16 million people. So we wanted to address this question, and we did this in collaboration with Brazil, and we were initially very interested in non-B subtypes because, of course, they're dominant in the non-Western world. And so we approached Brazil with a particular interest for non-B subtypes, and we tried to collect samples from people failing with dolutegravir, and then we sequenced their 3' PPT regions, and we looked for resistance mutations. the way the trial, this clinical trial was designed was actually flawed a little bit because there was no baseline sample collecting. So we still were able to identify series of 3' PPT mutations, and then to characterize them, we use methods that you saw a bit yesterday, whereby we can introduce them in models that we can transfect, and we can produce viruses that contain these specific 3' PPT mutations, and then we can test for their infectivity and resistance. So this is the only result slide that I have, so it can be relieved. These are the 3' PPT mutations that we found. We found these mutations in about 10% of people failing with dolutegravir. However, when we did the characterization, you can see here, this is a measure of the infectivity. So the higher it is, the lower the infectivity is. And this is in maximal infectivity compared to the wild type. And then you have the fold-change resistance against dolutegravir, which is here. And in green, what we found was actually often hypersusceptibility to dolutegravir inhibition. And there was only one virus that had minor resistance change against dolutegravir. And a lot of them were actually slightly impaired in viral fitness. So what does all of this mean? It's very simple, is that apparently these 3' PPT mutations are not, it would be still very important to study their prevalence at baseline in people who were not exposed to dolutegravir. And they are still so. And with this, I'll be happy to take some questions. Thank you.
Thank you very much for that insightful presentation. Please, we have time for 3 questions maximum. So.
Thank you, Thibault. Actually, when I was reading your abstract, I was quite surprised because I think a few years ago in Bordeaux, there was this investigational drug that when they do the drug selection, the resistance selection, they were selecting mutations in the 3PT region, and they saw that that was conferring resistance in the virus. So, I mean, maybe I haven't, I'm just wondering, how does this fit in line with that? I think it was Anne-Marcelin's group.
Yeah, so that's a good question. So that not all 3' PPT mutations are equal, essentially. So if you have deletions in the 3' PPT, including outside of this G stretch, and specific mutations in the 3' PPT in the G stretch, then most likely what happens is that you indeed go towards this pathway of 1-LTR formation, and therefore you can develop resistance. But apparently this is probably not a process that happens very readily in people, and that's why the mutations that you find in people versus what you can do in vitro or in cell culture, essentially the mutations are different. And I would be surprised to see the mutations we see in vitro in people, to be honest. Yeah.
Okay, thanks.
Any more, please? I can't seem to see any other. Okay, right here, please.
All right, thank you very much. I want to believe the mutations in the PPT region leads to or avoids or prevents the integration step from happening. How does this affect reservoir situation? So
You are and produced in a different process. But these 2 LTR circles can be found in people under treatment for more than 10 years. So before, we used to think that they are a byproduct of abortive infection and they are essentially garbage. But they are not. They are probably potentially another source of rebound. This has to be tested. But definitely the HIV reservoir seems to be more complex than just integrated DNA. Whether these 1 LTR circle forms are also part of the reservoir remains to be studied, because there is no way, or it's quite difficult to distinguish them from 2 LTR circles or from integrated DNA. So the molecular tools are not there quite yet to study this, but we're working on it. So yeah, we will try to address the question. Thank you. And thank you again for the kind invitation. And thank you, Peter. I see you're not listening to me anymore. No, I'm joking. So, but I'm also very happy to be here. Thank you.
Thank you very much.
Okay, that was indeed technical. We'll go to the follow session. We have our first speaker, Mark Apienin, who is currently characterizing the HIV cohort in Ghana. He's going to talk about his contribution towards HIV cure research.
Thank you very much. My name is Mark Apienin, and I'll be talking about characterizing an HIV cohort in Ghana and its contribution towards HIV cure research in West Africa. Having listened to a very broad talk and a very narrow talk, I hope mine will fit into the middle so that we can all flow with me. Thank you very much then. So HIV has been with us for the past 4 decades, but still continues to be a disease of public health concern, with about 39 million people living with the disease as of 2022. Number of new infections continued to decrease, but in 2022 we had 1.3 million new infections, with a little over 630 million HIV-associated deaths. In Ghana, Auditorium has a high genetic barrier, which we've heard before and, in our previous talk, and the fact that it doesn't require any boosting. And also, it is known to have limited cross-resistance to other first-generation integrated strand transfer inhibitor. Let me say that dolutegravir is the first of the second-generation integrated strand transfer inhibitor that was approved for use by the US FDA in 2013. Again, in Ghana, since with the introduction of the dolutegravir-based regimen, we do not have a lot of data on how well it is doing within our community. So it is instructive to know that the virus that causes the global pandemic, that is HIV group M, has a lot of subtypes, and these subtypes are distributed, are found within different geographical areas. In subtype C is the predominant subtype found within Southern Africa, whilst within Western Africa, what we call the circulating recombinant form O2 is the predominant subtype of HIV found within West Africa. In Europe and most of the Americas, we have what we call the subtype B, being the predominant HIV subtype within these geographical regions. And most of what we know about HIV has been on subtype B, because that is what is found in most of the most European countries and in the Americas. It is instructive to also know that there are differences in the genomes of these various subtypes, and these may have impact on the response to antiretroviral therapy. Indeed, it has been shown that some mutations, or these mutations have different impacts in different subtypes, or the integrates of different subtypes in culture. What then do we do? Having known that we have different subtypes in different places and may impact the response to HIV, it is instructive, or it is essential that as we look for new treatment, as we look for new cure for HIV, it should be geographically inclusive. And that is why we are embarking on this study. The rationale is that we believe that a well-characterized cohort in Ghana will be essential, as it will help us determine the impact of various drug regimen in Ghana. It will also provide us the opportunity to undertake cure research, cure-related research, including clinical trials. Then, more importantly, like I've said, most of what we know about HIV has been on subtype B. But what we have, the predominant subtype in Ghana, is the circulating recombinant O2. And this cohort will provide us the avenue to understand exactly the local epidemic. Some cohorts elsewhere have given us the opportunity to understand HIV. For example, this Capresa cohort in South Africa has given us a lot of understanding to HIV in South Africa. That is the subtype C, and a lot of clinical trials have been embarked on in South Africa because of some of these cohorts. Additionally, this also fresh cohort in South Africa has also given us the opportunity to understand the immunology of hyperacute infection in South Africa. So it is important we have something like that. So with this work, we aim to establish a long-term HIV infection cohort in Ghana. And to do this, we recruited people living with HIV and comprehensively characterized them. We also studied the cellular immune response within this cohort. Then again, we evaluated the impact of dolutegravir-based antiretroviral therapy on the viral load within this cohort. This is how we went about it. We obtained ethical approval from the Ghana Health Service and ECBAS. And within this cohort, we have two categories. We had the naive, ART treatment naive group, which we followed them up every 6 months. Then we had another group, which were the ART experienced, which we cross-sectionally sampled them. Then we have so far sampled from four hospitals, 1 in the Volta region and 3 in the Greater Accra region. We are looking to hit a target of 1,000, but we are not there yet. So we continue to expand. So the first part was to establish the cohort, and this is how we went about it. We recruited the participants being both ART treatment naive and ART experienced. Then we reviewed their medical record, administered questionnaires, we drew blood sample, we continued to do HIV typing, we did some screening for sexually transmitted infection, including hepatitis B, C, and syphilis. We did hemoglobin estimation, then we looked at their liver function, we looked at their renal function, as well. So far, we have been able to recruit 380 participants. We have 153 being ART naive and 227 being ART experienced. And these are the contributions from the various sites, the highest coming from the University Hospital here in Legon. By way of age distribution, we realized the majority of the participants are within the 15 to 49 year age limit. The implication is that these people would have to be on treatment for the rest of their life until we find a cure for HIV. And these are the working, majority are the working group. These are the modal responses for the various characteristics that we try to look at in trying to characterize them comprehensively. We have gender, we have the educational status, we have the occupation, we have the marital status, and we have the possible mode of transmission. It is instructive to know that within this cohort, the common transmission route is through the heterosexual, and this is most common in all other West African countries, as compared to the West, where it is through mostly homosexual. We looked at self-reported conditions, and we have 13.7% reporting of reporting having a chronic condition, hypertension being the most common. Then we looked at the ART regimen on which these participants were on. We realized majority, 97.6% are all on ART, the dolutegravir-based regimen. And out of those who were already on treatment, some had a change in regimen, and mostly it was because of the change at the national level. That was in 2019 when we adopted the dolutegravir-based. It's 0.5%. Within our cohort, I must say we have 13 pure HIV-2 participants and 44 who have dual infection. Anemia was prevalent within this cohort. So then in terms of, we looked at the trend, the response to, we looked at the response as, we looked at their responses by measuring their hemoglobin, and we realized there was increase in hemoglobin concentration from time as they started treatment. And we looked at whether there was a damage to their kidneys, but we realized that at the beginning we didn't see any damage. But with long-term treatment, we see there's a reduction in the eGFR. Then we looked at the liver as well. We realized there was no significant, there was no damage to the liver as we see. Okay, there was no association between treatment status and the and renal function, as well as the liver. So we say that we have been able to establish the cohorts, and we continue to expand on the majority willing to participate in any future clinical trials. The second part was to look at the cellular immune response, and to do that, we looked at the CD4, CD8 T cells, and we estimated them from whole blood using a two-color flow cytometry. We say that there was a significant improvement in the CD4 count after 6 months of treatment, and even to the recovery to normal levels within those who were ART experienced. For the CD8 T cells, we realized there wasn't significant differences for the treatment naive that we followed up, but between the decreasing with treatment, but it wasn't decreasing at the rate at which we expect to see. Okay, so we had a lot of participants still having viral non-suppression within this cohort. So in summary, we are saying that there's improvement in dolutegravir based. There's improvement in the viral load, but then it's approved or some verified kit for the typing. So in our case, we use the SD BioLine for the subtyping, for the typing. Yes.
All right. Okay. Thank you.
I'll spare that. Yeah. Please go ahead.
Okay, so there are a number of things that can be we can look at. It could be, we are yet to find out if there are drug-resistant mutations within the group. That could be one. Whether there are drug-resistant mutations in there, or there could be the fact that people are not adhering to the treatment, which is another arm that someone else is looking at.
Okay, I have 2 questions, okay, but I'll ask you later because your time is up. Thank you so much, Mark.
Thank you, Mark. We have our next presenter, Abigail Obobi, representing unveiling the Plasmodium inositol phosphate pathway, highlighting IPMK as a novel therapeutic target for malaria. Abigail, you have 15 minutes.
Thank you very much for having me here, and welcome to malaria. Okay, so we are back to malaria. So this work is part of my PhD work, and the foundation of my work is we know the problem with malaria now, that there's a development of resistance towards very effective antimalarials. So my vision was to find novel ways to target malaria. So I'll start with the pathway. There's an interesting pathway in eukaryotes called the inositol pyrophosphate pathway. And this pathway involves the sequential phosphorylation of inositol trace phosphates, popularly known as I3P2, inositol hexacase phosphate, and inositol octacase phosphate, known as pyrophosphate. The players in this pathway are kinases, and we all know kinases are well-known drug targets. And for the purpose of my presentation, I'll be more interested in the inositol polyphosphate multikinase (IPMK), inositol pentacase phosphate 2 kinase, inositol hexacase phosphate, and diphosphoinositol pentacase phosphate kinase. My work too is a bit technical. I'll try to calm down as much as I can. So this pathway is an integral part of calcium and phosphate or energy homeostasis. And if you know the biology of falciparum, the most virulent causative agents for malaria, we know calcium metabolism or calcium signaling is very important. especially in synchronization, which helps the parasite evade the host immune response. So having known this, I did a little background, and I saw that the pathway is essential in humans. This pathway is essential in other parasites like Trypanosoma brucei, the cousin of Plasmodium, which is the T. gondii. I've seen the pathway essential in plants, and one popular metabolite of the pathway is phytic acid. We know even as humans, some people take that thing as a supplement. So having seen all this, I didn't see this pathway being studied in Plasmodium. And this led to the hypothesis where we hypothesized that the Plasmodium inositol pyrophosphate pathway is present and essential to the the erythrocytic Plasmodium parasites. And to answer this, we had 3 objectives. Basically, we dwelled on the powerful tool of bioinformatics and PCR to be able to characterize the genes, putative genes in this pathway. We confirmed the presence and also in silico chemically validated them. So the summary of the methodology I used, we sought ethical clearance from the Noguchi IRB. Then went on to sequence and structural analysis, PCR with sequencing. Then we used molecular docking to do virtual screening to find selective inhibitors to the kinases. So we were able to identify 4 genes which encode the kinases in this pathway in Plasmodium falciparum. The first one we call IPMK1, then we have IPMK2, IP6K. Then the last one is actually a multi-domain gene which encodes an enzyme called VIP1. So with the architecture of the kinase domain, with the architecture of the kinase domain, we could identify several intersections in the Plasmodium enzymes as compared to that of the humans. And this kind of confers special activity or special functions to the Plasmodium, and it's popularly known actually in parasites. But with the sequence identity, we did a pairwise sequence identity, and we could see here that these enzymes actually, let me chip this in, they are broadly divided into 2 subgroups based on the functional motif. So we have the PDKG enzymes, and 3 of the 4 genes I studied were PDKGs. So I grouped them together here. These 3 are PDKGs. So you could see that they had very low sequence identity to the human enzyme. And the 4th one, which is the VIP1, had 44.7, which is a bit high. Then we proceeded to characterize them in terms of evolutionary relationship. So what I could simply show is that the protein, all what I'm dealing on now is actually the kinase domain, because I know when you talk about kinase domain, those are a bit specific than looking at the whole protein. So every analysis I did here, I used the domain, kinase domain. So if you look at the domain labeled in blue represents the Plasmodium PDKGs, and the pink one represents the human PDKGs. And we could see that the Plasmodium enzymes are analogous to that of the humans. And for the VIP, we could also see the Plasmodium, which is blue, that is the most diverged in the analysis we did. Then we went on to determine how structurally distant they also are. We know protein fold energetically, protein fold because of how energetically favorable they could be. So when checking the structural divergence, we modeled the structures of the 4 domains. Then comparing to the human, we know kinases have different parts. We have the C lobes, the hinge, the IP helices, where the substrate binds, and we have the N lobe. They are quite similar to the protein kinases. So when we did the okay, it's not moving. So we did the structural alignment, and the cutoff we used was the RMSD values, which it's not responding. The RMSD, yeah. So we did the structural alignment, and the cutoff we used was RMSD value of 2. So any alignment we got, the value we got, if it's below 2, it denotes homology. So we could see that both IPMKs here are structurally dissimilar and not homologous to that of the human, as compared to the ICSPK and the VIP. So the other important thing we did was to able to, because they are kinases, we know kinases bind ATP. So we wanted to now we have the same, two genes, which supposedly does the same thing. So we wanted to make sure that they are all okay. And as I said earlier on, in the identifying or in analyzing the sequences, we saw a mutation in the protein sequence of the IPMK1. So here I wanted to find out if structurally that mutation is going to affect the binding of ATP. So what we did was to dock ATP in. And we could see that we were expecting ATP to bind to the hinge region, which we predicted the residues to be valine and the glutamine. But the ATP took the binding, the mutation was able to take part in ATP binding, which is not supposed to. Meaning if this protein is even expressed or produced, it's not going to be functional. So we then moved on to, we moved on to the IPMK2, and we could see clearly that the adenine moiety of the ATP was able to form hydrogen bonds to its right predicted residues, which shows that the IPMK2 is not redundant. And it's essential because we've gathered data from the whole genome-wide mutagenesis studies, which shows that this gene is essential. So I now bring you to the pathway So this pathway starts from the cleavage of PIP2 by PLC, phospholipase C, to diacylglycerol and I3P. And we know from our classical biochemistry that I3P is involved in calcium mobilization from the intracellular stores. But IPMK is able to use I3P as a metabolite, which sequentially phosphorylates to I5P or IP5. And we have I6PK, which also is able to phosphorylate it further to a pyrophosphate. But what we saw in this plasmodium pathway is that it lacks the inositol pentakisphosphate kinase, which is supposed to phosphorylate I5P to I6P. So for this, we hypothesized that probably the parasite scavenges the IP6 from the host, or I6P is able to synthesize the substrate. And we know also from this, the I6P also behaves as an ATP synthase, where at low ATP is able to synthesize ATP from ADP. When ATP is high, it's able to, the pathway favors the pyrophosphate production. And this pyrophosphate also is implicating several signaling processes, as well as acting as a metabolic switch. So as in if ATP is low, the VIP gene has a histidine acid phosphatase, where it can cleave off one moiety of phosphate to release energy to supply the parasite. And in the nucleus, we could see it was based on data in literature. The pathway in the nucleus is involved in signal transduction, epigenetic regulation, and all that. So one of the most important things we did was, because we were dealing with putative genes, we wanted to find out if it's actually present in the parasite. So the F1 is the field isolate, 3D7 and DD2. We're able to find the parasites, the genes in there. So we went to the structure-based discovery. So we did molecular docking to find out if we could see inhibitors that have preferentially high affinity for the plasmodium enzyme than that of the human, and we could identify a few of them. Then here is the fragment-based discovery. So we started with a library with very small molecular weight ligands, and we were able to find a scaffold. So the initial binding here, we could see that it had good interaction with the enzyme. Then we did a substructure search in zinc and other databases, PubChem and all that. They were able to find some agents which had a bigger molecular weight, which also showed preferable activity for plasmodium protein than the human. So here, I know it's a bit technical, but what I want you to take home is that we've seen an interesting protein that is not reductant, it has a sufficiently unique structure, it's not autologous to the human enzyme, and it's druggable. And the advantage of pursuing this is that it has a possibility for selective inhibition. It has potential activity across multiple life cycles, and possibly for like a possibility for panspecies activity. So we have seen, we have evidence, we have shown the evidence of the presence of genes encoding the inositol pyrophosphate pathway in Plasmodium falciparum. And overall, our data shows that it shifts the signaling from I3P towards the synthesis of higher inositol, which are energy rich, and we believe that it's important in calcium homeostasis and phosphate homeostasis. And that if we target this pathway in Plasmodium falciparum, it may provide different or a new avenue for anti-malarial agents so that we overcome the evolution of drug resistance. Thank you. So here I acknowledge my supervisors, my Prof. Ben Kwashi, Prof. Nancy Kwashi, and Professor Sayers from the University of Sheffield, and all the lab team from Kwashi's lab and the Sayers lab. I'm very grateful for the support given me. Thank you.
Thank you very much. I don't know whether you succeeded in delivering on your promise of breaking it down, but the audience are in a better position to do that for us. So any questions for clarification? Yes, one there.
Yes, so my question is still on that. Can you please summarize your research to the watch sell at night market?
Okay. Okay, so basically, to the watch seller. Before, as Africans, I don't know of any Africa or any Ghanaian that have not had malaria before. And we know the effect of malaria. I didn't actually believe malaria could kill until I had a very close relation having a complications in malaria. I realized that it's deadly because I didn't understand why malaria should kill anyways. But once I experienced it, I know that it's deadly. So for the development of resistance, where we take in drug and it's not able to work on our system or kill the parasite, it will lead to more death. So my mission here is to look for new ways, which I feel like would be very important to the parasite. So if you take that thing away from the parasite, they are going to die. So the new ways I'm looking at to study the biology of the parasite and find agents that would inhibit or target that aspect of the very important to the parasite. And hopefully we can develop a new drug towards malaria, or against malaria, sorry.
Okay. Thank you. There's a question in front, please.
Actually, I was wondering whether you were referring to I3P or IP3 or NIP6. And then when I realized on slide 14, you actually put I3P.
Yes.
Should it be IP3?
Yeah, the nomenclature of these agents are very confusing. In literature, some people say I3P.
It's inositol.
It's inositol trisphosphate. So I feel that's why they write I3P. I usually pronounce it IP3. But when during my literature search, I realized they more use I3P, I4P, which even the pronunciation is difficult.
The way you write it means is you have 4 or 3 molecules of the inositol to 1 phosphate.
Yeah.
That's what you have written.
Yes. So as I said, that's what is in literature. But I felt it had to do more with the nomenclature, how they pronounce it. Like inositol tetrachisphosphate, inositol trisphosphate.
Please, one last one at the back.
Thanks, nice talk. Whether it's tomato or tomato, something to think about is even though it doesn't have targets theoretically in, let's say, human systems and cells with this particular signaling pathway, something that's worth doing as part of your work is stimulating some human cells with it and looking at some cytokine, chemokine profiles by hitting it with something perhaps like hemazone to just see whether or not it changes those profiles. So some of these drugs, despite their specificity, alter the immune profile in the human cells, thereby changing some of the expected actions. And it's a straightforward experiment to do, and I'd encourage you to do it. Thank you.
Thank you, too. Well noted.
Thank you very much, Abigail. Let's appreciate her.
So we managed to survive technical sessions. Yeah, I had a lot of questions, but I don't even understand the questions myself. So I'll just keep it. Thank you so much, our speakers. Please give it up to them. So our next session is the table talks, and that will be led by Dr. Peter Kwashi.
Let's thank Dr. Bonnie and Dr. Arthur for a great session chairing. So those of us who follow us on social media would have seen that we do a number of events during the year. And one of the things that we did few weeks ago, maybe 1 or 2 months ago, was that we had this vaccine science forum, and then we had a 2-day open science day here at WACCBIP where we invited 7 high schools to come, and basically we exposed them to WACCBIP, what we've done, our impact. They went around the labs, they did some little experiments here and there were some demonstrations, and then we gave them a challenge. If they have some ideas, innovative ideas, they should send us those ideas and then make a poster of it that we'll present here. So we have students from 2 schools who will come and do a turbo talk to present the idea that they had. I haven't seen the idea, so I actually can't tell you what it is. And they're from the West African. Have they entered here yet? I'm preempting them a little bit. Yeah, but there will be students from the West African Senior High School, WAS, as is popularly known in Accra, and also students from Frafraha Community-Based School. So they have their posters on the first floor. When you walk in, it's up against the wall so you can see it. And this is our way of trying to encourage these up-and-coming young scientists, we hope. So they just arrived here from school with their school bags. So unlike, they don't have a one slide, but they'll come up and talk about their poster and invite you to their poster. So I'll ask the students from West Africa Senior High School to Senior Secondary to come up. Was, come and talk about your poster. And this is in the same manner. Oh, come. So you introduce yourself and you tell us about your poster and why we should come to it. And this is in the same manner as it's a turbo talk, basically 2 minutes.
Good evening, everyone. Please, I'm in the person of Emanuela Amanyako Nsiedu, and I stand before you today not just as a student, but as a survivor. A survivor of malaria. Growing up in Bonata, so that concussion recovered me. So it was then I realized the potential power of nature to heal. So it's artemisinin-based combining therapies, ACTs. ACTs. Her story ignited me. Her story ignited a fire within me. proving that even a woman from a humble background can make a significant impact. So that was where I got my inspiration. So Ghana, my beloved country, has eradicated anti-malarial drugs in order to make a malaria-free future for us. Thank you.
All right. So let's give her another round of applause. I think nerves are something that all of us experience, but she did well. Notwithstanding. So after that, I'll call Frafraha Community-Based School to come and present their idea.
Good evening, everyone. Okay, so I'm with Bolabi, and then I'm here with my colleagues to talk about a virus which has affected over 80% of the worldwide population at a point in their lives. So it's a leading. This virus is a leading cause of cancer in our daily life. So I would like someone to guess the type of virus I would like to talk about. Yes. So I'm referring to the human papilloma virus, which is commonly known as the HPV. So I would like to talk about a short story which inspired us to go for the human papilloma virus. So my auntie in her mid-30s was diagnosed of the human papilloma. She was diagnosed of cervical cancer. But fortunately, she received treatment and she's cancer-free now. So her experience and the pain she went through made us want to talk about the HPV, its importance, yes, and the impact it has on our lives. Even though the HPV is very common, it is often misunderstood by a lot of people around us. So I would like to talk about I would like to give a detailed presentation of the HPV when you come over to our poster. Yeah, thank you.
Okay. So we are let's give another round of applause. And then we're going to call people up to come and give their turbo talks, which is a one slide. It's not every poster that got a turbo talk. So these were posters that were highly scored, and so they'll come and give a one slide and talk about why we should go to their poster. While others are being lined up, I'll call up the first two, Gloria Pokuamenu and Ernest Afari. And you have 2 minutes each.
Good evening. Okay. My name is Gloria Pokuamenu, and I'm presenting a poster on the topic predicting SARS-CoV-2 antibody binding to selected respiratory viruses. This is to explore the potential for cross-protection. So respiratory viral pathogens are a public health concern, and they cause respiratory viral infections. And a recent report on the leading causes of death shows that respiratory viral infections are the second leading cause of death in Africa. And this is quite an interesting report, because the morbidity and the mortality of respiratory viral infections in Africa is quite high. And so what is the problem? The problem is that there are no licensed routine vaccines for many of these respiratory viruses. And currently, only 2 vaccines are licensed for routine use: SARS-CoV-2 and influenza virus. Just this July, 1 vaccine was licensed in the USA for respiratory syncytial virus, but even at that, it's only for the aged. And so we asked the question, could the impact of COVID-19 have any beneficial effects for us? Looking at the extensive vaccination that has gone on globally, could we harness the benefits for control of these other respiratory viruses, since it is very important to look at alternative control measures? And so in this study, we tried to use in silico analysis to bind the antibodies of SARS-CoV-2 to other selected respiratory viruses. So for my poster, what we did was that we showcased the results for respiratory syncytial virus, and the results are quite interesting. We first looked at aligning the antibodies for the 2 different viruses to see if there are similarities or identities between these antibodies, and then went further to dock SARS-CoV-2 antibody with antigens from respiratory syncytial virus. The results that we obtained is quite interesting, and I would love that you come to poster number 13 and for a detailed description of what was observed in our study. Thank you.
Hello everyone, greetings to you all. Okay, I'm Ernest Afare, presenting on Plasmodium falciparum antigen-specific antibody levels and avidity of antigens in Ghanaian individuals across varying malaria transmission intensity. Humoral immune response are key in clinical immunity against P. falciparum specific antibodies or infection. In high malaria endemic regions, affinity maturation of P. falciparum infection is hampered in both children and adults. This study has been done on the blood stage antigen, which is the MSP3. It is unknown if antibodies against the pre-erythrolytic stage and the sexual stage antigens are similarly hampered in high transmission settings. So the study estimated antibody responses and avidity of antigens from the 3 different stages of P. falciparum specific antigen. That's the liver stage antigen, CSP, and the blood stage antigen, MSP3, and the sexual stage antigen, that's PFS 230. We use an indirect ELISA method. 316 plasma samples were used, of which 174 were from Accra and 142 were from Cape Coast. Accra is demarcated as a low malaria transmission region, where Cape Coast is demarcated as a high malaria transmission region. Come to poster number 5, and my finding will shock you. Come listen, come and be educated, come and be imparted. See you at poster number 5. Thank you.
All right, thank you, Ernest. So I'm waiting for the next few slides to be queued up. So Chris, give me a thumbs up when you're ready. In the meantime, let's get up and do some stretches. I know we'll be walking soon, but let's take advantage of this. We've been sitting for a while. And so, okay. All right, okay, so we're good to go. So let's get ready. I'm going to call up the Turbo Talks, Becky Eramatete. So you can line up, and once the first one finishes, the next one goes. Becky Eramatete, Precious Kwesi Kumi, Timothy Letza, Yinka Submarine. And then we'll take these for now. So I didn't realize they reordered it. Okay, Ernest, we've already. So Precious Kwesi Kumi. Is Precious here? That's not your slide. Theo, that's not your slide. No. Precious, you stay. So Precious Kwesi Kumi, Timothy Letza. Let me see. 1, 2. Lucinda Frainate, Eugene Lamte. Can we have slide number 1? Huh? This slide number 2. Come and talk to me. Come in. Okay, so let's do it like that. If you see your slide, come up and talk about it. I mean,
Hi everyone. Okay, so just imagine you're a bacteria, and then you have coat, and then you have different colors of this coat, okay? And whenever you get to wear one of these coats, you get to have a different identity, and that identity gives you a different level of causing harm without being caught. If I were you, I will always put on that coat, right? So I'm working with Streptococcus pneumoniae, which is a gram-positive bacteria, and I'm looking at how 8 coat, which is a polysaccharide capsule, together with 8 antimicrobial resistance, is helping it to evade its host without being caught. My poster is poster 4, and my name is Jennifer Morgan. So please come and hear more about that. Thank you.
Hi, I'm Precious Kumi. And I'm here to talk about COVID-19 booster immunity. Basically, how long did your booster immunity work? That is with respect to the COVID-19 vaccination that you received. Now, booster vaccinations will typically stimulate your immune system to generate a heightened response against COVID-19. Now, the quality of that response is affected by a number of factors, such as your immune status, your previous vaccination history, and then to a large extent by maybe the type of booster or the vaccination regimen that you would receive. Now, I'll be looking to measure T cell and antibody responses against, I measured T cell antibody responses against COVID-19, and then the goal was to determine how long your immunity lasted. So please meet me at poster 3, where we can have a lot more conversation about this. Thank you.
Good evening, everyone. I'll be presenting on cytokine profiles in asymptomatic malaria children with different hemoglobinopathies. Okay, so asymptomatic malaria remains a public health concern, particularly in regions where it is endemic and prevalent. This is because individuals who have this condition do not report the disease, and they contribute to the transmission of the disease since they don't report it. And also, genetic factors genetic factors like sickle cell disease are also associated with inflammatory responses, and these inflammatory responses are mediated by cytokines, which are proteins and soluble molecules that regulate the immune responses. So in my study, I measured or I assessed the levels of pro-inflammatory cytokines and also anti-inflammatory cytokines. IL-6, IL-8 for the pro, and also anti-inflammatory cytokines, IL-4 and IL-10, using the sandwich ELISA method. The samples were collected from 3 communities, Begro, Estuary, and Tomefa. So my study found out that individuals who were positive for malaria had significant levels of IL-10, indicating that indeed these individuals may be asymptomatic. And also, my study found out that individuals who are positive for sickle cell disease may be in crisis because they have significant low levels of the anti-inflammatory cytokines, which is the IL-10 and then the IL-4. Overall, my study contributes to the understanding of So you can come and meet me at poster number 15 so that we can delve deep into this. Thank you.
All right. Good evening, everyone. Yeah, so I'm trying to model malaria drug resistance, and yeah, so as we can see, malaria is a threat, as we already know, and it is expected that if this drug resistance that we are having currently facing, for example, the ACTs that are majorly used for drug resistance, if we don't look at this problem and how fast it will spread, then we shall have issues. For example, it's confirmed in East Africa, South America, and Southeast Asia. So what are we trying to look at? We are looking at linked populations of mosquitoes and humans, and we're looking at probabilities of events happening within the transmission dynamics of malaria. The major assumption we make is, what if it's easier for these drug-resistant parasites to pervade a population of people that are susceptible? What if it is easier for for the drug-resistant parasites to invade a population of people that are susceptible than those that are already carrying one mono infection? So we ask ourselves a question: does this assumption help us to understand the fact that resistance will spread faster in low transmission areas than high transmission areas? If you want to discuss more about this, I'll be waiting for you on poster 35. Thank you so much.
Hello everyone, my name is Lucinda and I'm presenting, my poster is on the impact of amino acid polymorphisms in Ghanaian HIV-1 post sequences on their structural characteristics, and this is an in silico study. So I'm sure we were all surprised and a little scared about the reports that was given in the news on the number of people infected with HIV in Ghana. So to jog our memories a bit, to give you that perspective, in 2022, Ghana recorded over 16,000 new cases. And these infected Ghanaians may resist antiretroviral therapies due to mutations in the HIV genome. So this study aimed to examine the impact of these mutations on proteins encoded by the HIV POL gene, which is integrates reverse transcriptase and protease. These are very key proteins that help the virus to survive and proliferate or thrive. Yeah. So we first retrieved the sequences from the Los Alamos HIV databases. So these are Ghanaian sequences. We used the Stanford HIV drug resistance database to perform a genotyping to identify the most prevalent mutations in these Ghanaian sequences. We then modeled these proteins and identified the effect of the mutations on their stability. We then did a molecular docking study with the known inhibitors of integrase, protease, and reverse transcriptase. And we then, so the molecular docking study was to see the change in the affinity for the proteins after these mutations have occurred. So we then move on to assess the interactions, the protein ligand interactions between the inhibitors and the mutated proteins. And then we do a molecular dynamic study to check the stability when these inhibitors bind. So meet me at poster 41 and let's talk. Thank you.
Good afternoon. My name is Eugene Lamte, and I'm presenting on a drug discovery project using machine learning. I'm presenting on a drug discovery project using machine learning targeting the herpes simplex virus, specifically the virion protein 16, and these machine learning models were evaluated on various machine learning various evaluation metrics such as the accuracy and other. Yeah. So different machine learning models had different evaluations because they have different ways of making classifications. And we were able to take the 3 best models and then deploy them using web servers. So at the stand, we would be able to give a live experience of how this, how the web server works, and use the models to make predictions on new data. Thank you. Poster 55.
Hello everyone. So my name is Caleb and I'm presenting on determination of virulent gene diversity of Helicobacter pylori in Ghanaian patients with gastric dyspepsia. And this study was done here at WACCBIP. Okay, so H. pylori is a gram-negative bacteria that contributes mainly to gastric dyspepsia, right? And then in certain regions, genetic variability of virulence factors play major role in the disease progression, or differences in clinical outcomes would be seen in Ghanaian patients with gastric dyspepsia. So what we set out to do was to investigate genetic variability in certain virulence factors of the Helicobacter pylori in Ghanaian patients, right? And then what we did was we in this study we took stool samples from the patients with the gastric dyspepsia, and then through targeted PCR amplification methods and then sequencing techniques, we amplified the Kage 3' variable region, and then we sequenced it, and then we did a multiple sequence alignment. So that's what we can see here. And then we had surprising results also. So please, if you want to know more about our findings, you can visit poster 73. Thank you very much.
Good evening, everyone. My name is Helen Kamasa. Then I'm presenting on this topic, bacterial vaginosis and correlates of cervical cancer in apparently healthy Togolese women. I'm happy that my work has been briefly introduced by these ladies here. So I want just to let you know that apparently healthy women can have anginal cervical cancer. And this cancer is the second most common cancer in women in Africa here, and it's caused by persistent HPV infection. But what is amazing is this cancer is the most preventable cancer, suggesting that there are other factors that are included or that are included in his risk factors. So you are kindly invited to visit me at the poster 77 for more interaction about my work. Thank you.
Hello everyone. My name is Becky and my work is on the functional characterization of H2 receptor in COVID-19 infected West Africans. COVID-19 pandemic is over, but it's still necessary for us to understand the dynamics that happened during the pandemic. And beyond the research that has already been done in malaria and looking at immune signatures, my work focuses on looking at host receptors that may have polymorphisms that may have led to the clinical phenotype for West Africans, which was predominantly mild. So we are hypothesizing that if people have mutations in ACE2, there's a probability that it may have a lower binding affinity to SARS-CoV-2, and that may lead to either severe, mild, or asymptomatic clinical presentation. And I have tested this using in vitro and in silico assays, and I would love to see you at poster number 83 to talk more. Thank you.
So, good evening, everyone. My poster is addressing the issue of antimicrobial resistance, and I'm so glad that Professor Basiro mentioned and talked about the One Health approach. AMR is a global challenge, and it's caused a lot of death. In Africa, it's estimated that about 250,000 people died in 2019 as a result of antimicrobial resistance. And to address that, a UN high-level meeting is happening in September to show you the importance that the global community attaches to antimicrobial resistance. A One Health approach is important. There have been a lot of work around human health and livestock in addressing the issue with limited attention to the environment as well as food. And food safety is quite important and could be one of the important means of transmission. We looked specifically at the fresh produce here, vegetables, especially those that are not cooked before consumption. And we saw some interesting reports in the sense that some of the pathogens, according to the World Health Organization, they released a priority list of pathogens for antimicrobial resistance this year, 2017 first and updated this year. And some of the isolates that we found were on that list. So if you want to find out more, please see me at poster 113. Thank you very much.
All right. Let's give some applause for those who did their. So once again, we're trending early. And what we did this year, in an effort to fit the schedule, is that we've combined the poster session with the coffee break. So you can get your snacks, take a look at the posters, talk to the presenters. But before we go, because we are going a little bit ahead of schedule, if you have your poster up, and we'll do this for the first 10 people. If you have your poster up, but you are not a Turbo Talk person, and you want to give us just some 2 quick things why we should come to your poster, you can come up right now.
Hello, everyone. My name is Richard Opoku, and I'm presenting on the topic Genomic Characterization of Klebsiella pneumoniae Isolates from Komfo-Anokye Teaching Hospital. It's about isolates that have been gotten from clinical samples, and we are going to use MADITOF to confirm their identity. Thereafter, we will do sequencing. So I need you at stand 119. I hope to see you there.
For the posters. Okay, yes. To prevent the mix-up, for tomorrow we'll have another Turbo Talk session for the posters. So the Turbo Talks today are for the posters which are already up for today and tomorrow. Tomorrow we'll have another Turbo Talk session for the poster sessions Thursday and Friday. So when you are submitting your slide, make sure you mention the day that you are presenting and your poster number, which you'll get from the book of abstracts. So hopefully we'll avoid this tomorrow. So I'm going to do the wrap up for today. I think we had quite a good round of talks, more technical and some a little bit specialist, but I think we appreciate the breadth of expertise and knowledge and research that is being presented here. Tomorrow we'll kick it off with a keynote from Professor Herman Sintim, and that will be even more technical, talking about drug discovery, but that will lead into another session on immune responses and disease pathogenesis. Then we'll have in the afternoon session, we will have a change from the schedule. Our second keynote of the day will actually be Professor Nikes Ndam, because he has to catch a flight the next day, and he will switch with Dr. Janelle Binman. So I think tomorrow is going to be another very strong day of talks. In the meantime, I think we can wrap up today. Let's go have coffee break. Enjoy the posters. Please do not go home right away. As much as we wanted you to have a bit of a break, don't go home right away. Enjoy the poster sessions and any other housekeeping items. Andrew. Yes, and don't forget to go to the vendors. I'm sure they have some toffees or something. And then we have the WACCBIP shirts, the WACCBIP material already sewn. Okay, we're done for today.